Hello again. I'll start off the series of videos by looking at the first file, which is 31401. I'll try to provide you with this one because for this one I've highlighted some facts that are making it easier to read, but I'm not sure whether sometimes the highlights do not get transferred. But anyway, so if one of the first things you will see in this paper is that there's quite a lot of terms or phrases that have been put in bold. So that would be a start for you to have a look at the terms and make sure that you understand what each of them says. For example, in this the first box is just an overview of the whole chapter, which talks about what is a mutation, what are gene mutation, extragenic mutation. So along the way in the text they will give you additional explanation. So it starts off with looking at what is looking at the structure and function of structure and functional consequence of mutation. It starts off with the structure. I'll focus on the tables because again quite a lot of the explanation are in the text. So basically, in terms of structure, they are categorized based on the extent of the mutation. Either it's a point mutation where it involves one or several bases up to 5, and then you have complex mutation, macro mutation, and as well as then total chromosome imbalance. And then it also gives you an idea of how do you get this mutation. Like for example, point mutation can be caused by spontaneous errors, errors in replication. Or you have replication of a template containing a single base misinstructional lesion. Again, as I always mention, do not get intimidated by terms. Misinstructional lesion. A lesion means is a damage. Misinstruction, I mean, it gives it a wrong information. For example, when you have remember the G, if it can change the property of a G to bind with a T, for example. So that is an example of a misinstructional lesion. So damage or inappropriate base, so that can cause point mutations. Replication of a template containing a single base, non-instructional. So the difference between misinstruction, it gives a different signal or wrong signal. Non-instructional means it doesn't give any signal. This is probably, for example, the abasic site. The apurinic or apurinic doesn't have a signal. So polymerase cannot put in the correct base. So these are the causes of base substitution. If you go along the way and you find some terms that you're not familiar with, please don't hesitate to ask me. Send me a text or ask me in class, whichever. And then multi-site mutation, complex mutation. Normally you'll find this where you see repeated mutations. This is probably one of the causes because your mismatch repair system is faulty, or you have low fidelity DNA polymerase. What is low fidelity? That means your polymerase is not copying very well. It makes a lot of mistakes. Low fidelity. High fidelity means when you copy, it's almost the same as the original. So that's high fidelity. Have you heard of the term hi-fi? Hi-fi, hi-fi system, where you talk about your radio system. It says a hi-fi. Previously, there was this hi-fi. That means you can listen to music like you were in a concert. So that's why it's high fidelity. It repeats the sound of the concert in the radio. So that's why high fidelity. So low fidelity DNA polymerase means DNA polymerase makes mistakes. And this error-prone replication, if you remember in your, if I had taught you your second year, then we towards the end of molecular genetics course, there will be looking at the repair systems. One of the repair systems that will be introduced there is error-prone. Error-prone is a system where it just tries its best to repair DNA damages, and it's error-prone. I mean it makes a lot of mistakes. So these are some of the causes of complex mutations. And then macro mutations. affects single site but affect many consecutive nucleotide residues. It involves deletions and additions and amplification. So it's a lot of the texts are self-explanatory. My only worry is that you come across some terms that you're not familiar with. Again, let me know if you have any problems. Why do you get macromutations? Because of problems with chromosomal breaks and random rejoining. This occurs probably during crossing overs. You have homologous recombination between non-allelic sequences. That's where it's not supposed to be. Unequal crossing over, unequal sister chromatid exchange. I'm assuming you understand these terms. Recombination between dispersed repeats. If you don't, let me know. Site-specific recombination, we'll look at that in the combination topic. Mobile genetic element transposons. I'm not sure whether you're familiar with transposons. These are, we talk about Barbara McClintock, then we talk about transposons. So these are some of the causes of macromutations. And then finally we look at chromosome imbalance. We're talking about either aneuploidy, that means problem, or abnormal chromosome number, and also polyploidy, where you have double or triple sets. Polyploidy is normally acceptable in the plant world, but not in humans, right? Okay. Why do you get chromosomal imbalance? Is because problems with DNA replication system, where you're supposed to, the chromosomes are supposed to divide, mitosis is supposed to be followed by cytokinesis, and cytokinesis doesn't occur, so you have doubling of chromosomes, but the cells do not divide. Unscheduled or absent mitosis and meiosis, failure of chromosome segregation, fertilization. So these are some of the causes of chromosomal imbalance. The rest, later the explanations are given in text. After looking at a structure, it will look at the functional consequence of mutation. It starts off with giving you the general principle of functional consequence. Whether or not a mutation gives a problem very much depends on the mutation site. So when you talk about mutation site, you'll be talking about genes and related sequences and non-genes. So basically, even if you have mutations on gene-related sequences, you still are not sure that it's actually going to have an effect, because here this sentence says whatever the consequence of mutation per se, whether it's in the genome, coding sequence or not coding sequence, and whether these effects are expressed at the level of phenotype. So sometimes you can see changes in the protein structure, but you don't see the phenotype change. We'll look at some of the, there are some examples given in the text. So whether a mutation manifests itself as something that you can see, and something you can see. Okay, let me give you an example. If there's a mutation that changes your eye color, it doesn't give you any overall problem with fitness. You're still okay, correct? If you have a mutation that from a person who can roll your tongue, you become somebody who cannot roll your tongue, but it still doesn't give you any problem with fitness. So that kind of mutation, you would expect to see phenotype changes, but not necessarily fitness. So in most cases, when we talk about the effects of phenotypes, we normally refer to the fitness, right? So when we talk about negative effects of phenotypes, normally we talk about fitness, not just the causative phenotype, because some of them may be neutral, doesn't have any effects, and some of them may even be beneficial, depends. So whether or not the effects are expressed at a level depends on dominance, whether it's gene dominance or what is the genetic background. The word penetrant, you should remember what this means. I'll probably ask you in class later. I have to skip. This is number 2. So number 3 is down here. Mosaicism, right? Whether the population of cells, the one that has the mutation and the one doesn't have the mutation, what is the population in your body, because you know, if you remember the Luria-Delbruck fluctuation test, the theory is that cells divide. So if a mutation occurs like right at the very beginning, after the one, let's say one cell becomes two cells, then one mutates, right? But so after many generations, you have a population of normal cells, you have a population of mutant cells. So the phenotype also depends on the population of the cell. You know, the overall population of which one is you have, which ones do you have more. And then you start talking about monoallelic expression. Then you want to see whether which allele is actually expressing that has an effect. You know? So it's not as easy as, oh, there's a mutation and so you'll see an effect. But you know, but there are a lot of other considerations. Okay, I will continue in the next slide. All right, see you later.